My name is Michael, and my project investigates whether black flies can spread a cancer-causing virus to horses. The virus in question is called Equus caballus papillomavirus type 2, or ACPV2. It's a DNA virus from the Papillomaviruses family that specifically infects horses and is often linked to the development of genital as well as throat cancer in horses as well. Like its cousin, the human papillomavirus, it's most commonly thought to be a sexually transmitted disease. However, horses without breeding history nor contact with one another still frequently become infected with this virus. The virus will first enter via a wound or microabrasion in the skin, where it will target basal keratinocytes in the basal layer of the stratified squamous epithelium, which is just the outermost layer of the skin. Once the virus inside the cells, ECPV2 enters a latent stage where viral genome exists in an episomal form, meaning it's outside the host genome. During this phase, for the virus is not actively producing viral particles, and viral DNA replicates along host cell DNA. As the infected cells move upwards throughout the layer of the epithelium, the virus will become transcriptionally active, producing viral proteins as well as replicating its genome. In the uppermost layer of the epithelium, the viral proteins will form complete viral particles called virions. Virions are the infectious form of the virus. And as the infected cells reach their outermost layer of the epithelium and they die, they release the newly formed virions on skin flakes. As these virions get released into the environments, they can infect new cells, be transmitted to other hosts, or reinfect the same cells. In the case that the virus incorporates itself in the genome, the integration of viral DNA in the host genome disrupts the normal regulation of the host cell genes, particularly those in control of the cell cycle. This is done via the continuous expression of viral oncogenes E6 and E7, which creates neoplasia. Neoplasia is just uncontrolled cell growth. This causes a type of cancer called penile squamous cell carcinoma, or PSCC, which is a type of skin cancer that originates from the squamous cell from the stratified squamous epithelium and is primarily found in the genital region of horses afflicted with ECPV2. Tumors are often cauliflower-like and appear red with crusty surfaces, as you can see in this picture right here. So how do horses become infected if they don't breed or have contact with one another? We hypothesize that biting insects may serve as mechanical vectors for the transmission of ECPV2, as flies can bite into the epidermis of infected horses, potentially picking up ECPV2 virions on their mandible or legs, and when the insect bites into another horse, it creates that microabrasion or incision in the skin, giving the virus access to the basal cell layer, allowing infection of a new host. So in total, we had 6 cages, around 50 to 60 flies per cage, totaling 324 flies. We provided these 324 flies with an ECPV2 positive feeding substrate. The insect cage will also contain a spatially separated ECPV2 negative substrate, and we let the flies interact with a positive feeding substrate for 2 hours, and then after a period of 12 hours, we'll test the feeding substrates and the flies via PCR or polymerase chain reaction. So as I said before, the flies were first introduced in the positive cage for 2 hours, where they were allowed to interact with a positive feeding station as well as the cage that was smeared with ECPV2 positive tumors. After 2 hours, they were allowed to fly into an ECPV2 negative feeding cage where there was no ECPV2 in there as well. After a 12-hour period, we will euthanize the flies as well as test the flies, the cage, as well as the feeding stations to see whether ECPV2 transmission has occurred. And so, in order to measure while black flies retain the virus, we use something called qPCR to amplify specific viral DNA and measure the fluorescence of each cycle, so amplified DNA product can be quantified in real time. The fluorescence, or the dye, emits more signal as more double-stranded DNA accumulates. So, we decided to target the ECPV2L1 gene. As you can see in this amplification plot of our positive controls, that all of these crosses over this red line right here, indicating that there is amplification of ECPV2 DNA. For our negative controls, or our unknown samples, we saw that 2TE and 2TE, which are 6 flies collected from the negative feeding station in cage 6, both crosses over the CQ threshold, indicating potential amplification of the ECPV2L1 gene, indicating that they did have viral DNA on them. We also did a melt curve analysis, as similar melting curve values means that PCR products are the same with no major nonspecifics or primer dimers, indicating that the flies retained the true viral particles in their body for an extended period of time, indicating that transmission of ECPV2 has occurred via black flies. We also saw amplification in the samples at the bottom of cage 2, as these samples had the most time to interact with the virus, since these flies did not go up to the negative feeding cage and stayed in the positive feeding cages where most of the tumors were. They also had a similar melting curve to positive control. Overall, we wanted to clarify how ECPV2 transmission actually works, as it not only improves control in horses, but also provides insights into how other papillomaviruses, such as bovine and human papillomaviruses, might spread, as well as provide better vaccination strategies.